Hi, my name is Alice Maher and I'm looking at the evolution of body shape, locomotion and ecology in terrestrial vertebrates. So we know that vertebrates exhibit a wide range of diversity in body shapes, and identifying the factors that drive evolutionary changes in the body shape of vertebrates, however, has been a long-standing challenge because of the difficulties in sampling taxa broadly, quantifying complex morphologies, and identifying possible mechanisms responsible for generating macroevolutionary patterns. Most previous studies on body shape in vertebrates have focused on research into squamites or semi-aquatic vertebrates, but it is largely unknown how body shape has evolved more widely across terrestrial vertebrates in concert with changes in behaviour, locomotor style and ecological niche. Now, given the relationship between phenotypic similarities created by convergence, the use of phylogenetic comparative methods has therefore become almost imperative on analysis of evolutionary shape changes. Here I use three-dimensional computer models of vertebrate skeletons with phylogenetic reconstructions to quantify the evolution of body segments and whole body shape across 420 taxa of all major extinct and extant terrestrial vertebrate groups. So, the overarching questions I want to look at. The first one is a linear fit a better fit across all tetrapods, or is a quadratic fit better? And what I mean by that is most analysis of skeletal measurements against body mass show positive isometry, and these relationships have helped to predict body mass estimates in extinct animals such as sauropods by measuring the limb length of a fossil, for example the femur, and plotting it against extant species alive today. However, recent studies have reported lower body mass estimates for some of the largest extinct forms, the sauropods, and suggest that extrapolation of the linear relationship may lead to an overestimation and suggest that the relationship is log quadratic rather than log linear. The second question I wanted to look at is how do different locomotor and dietary categories scale, and are they different to each other? So there is very little literature on how different locomotor and dietary categories scale regarding allometry of skeletal measurements on body mass. Do we need to know more of the animal's origins to be more accurate in predicting the body mass of an extinct species? Or do different locomotor and dietary categories show different adaptations to the way species live as size increases? The third and final question I wanted to look at are different locomotor and dietary categories statistically different in body shape parameters. We know that tetrapods exploit diverse terrestrial and secondarily aquatic habitats, and central to this is body size and shape, which allows specialisms in a diverse array of habitats. Diet also has a fundamental impact on body size and shape, particularly in the skull, neck and torso of many species of animal. And studies into the fundamental morphology of a species' locomotor and dietary systems is therefore key to understanding a species' evolution. So we went about answering these questions by looking at a number of body shape and size metrics, which were quantified from 3D skeletal models of amphibia, aves, dinosauria, mammalia, reptilia and ancient reptiles, Reptilia morpha. We used whole body scans of specimens, which included CT scans, photogrammetry and online digital repositories. To examine patterns of body shape variation across terrestrial vertebrates, we derived a variety of linear and volumetric measures of body segment proportions, and these included the humerus, the forearm, the metacarpal and the manus, and the femur, the shank, the metatarsal and the pes. Now we know that body mass is the gold standard metric for overall animal size used to judge allometric changes in shape and body segment proportions. And many extinct organisms show good skeletal preservation, but the poor preservation of soft tissues prevents body mass values being directly preserved. Therefore, it's really important to find ways to accurately proxy body mass amongst extinct organisms. So what we did was body shape and body segment masses were proxied from reconstructions of gross morphology built around digitalised skeletons using a volumetric based method called convex hulling, which you can see in figure 4. This yields an approximation of the skeletal volume of each individual body segment in our models, which can be summed to generate a whole body skeletal volume. This is our chosen metric for overall body size, against which allometric trends in body shape and body segment proportions are judged. So to analyse phylogenetic trends in body proportion over time, each tetrapod species was added to a phylogenetic tree, which was built by merging recent taxon-rich cladograms of major tetrapod groups, and you can see that over onto the right. First and last occurrences of each species was found using the paleobiological database and the recent literature. This data was then combined with the phylogenetic tree to produce a time-calibrated tree, so we have species from the Permian all the way to the Quaternary. Locomotor and dietary categories were based on the primary mode of a species from the current literature, and Pagel's lambda was used to estimate the strength of the phylogenetic signal in the analysis. Now, it's not possible for me to go through all my results in the next 10 minutes, so I've picked some of the most interesting things that I found so far. Okay, when looking at is a linear fit a better fit across all tetrapods, or is a quadratic fit better? From our results, negative velometry and isometry dominate, which is consistent with an overall pattern of decreasing locomotor ability with increasing body size. Negative velometry is found more in the hind limb and distal limb segments. The forelimb is more isometric, which perhaps reflects that larger taxa become quadrupedal. We also find that quadratic fits are nearly always statistically significant, and in 3 cases they are a better fit than linear. This suggests that perhaps the largest taxa, so elephant size and above, are scaling differently to medium and smaller sized taxa. Again, this is consistent with studies on locomotor performance in elephants and biomechanical models of large dinosaurs, which suggest limited, if any, running ability at all in these animals. The second question we wanted to look at, how do different locomotor and dietary categories scale, and are they different to each other? So using phylogenetic ANCOVA, one of the interesting results we found was regarding skull size. So carnivores scaled significantly differently to all other categories when it came to the size of the skull. All slopes were found to be less than 1, with carnivores having the highest slope value. It has been suggested that larger carnivores have relatively bigger skulls as an adaptation to being large prey specialists. And although previous work has indicated that skull shape offers insights into function and ecology among a number of carnivore families, overall relationships between form and diet have been shown to weaken when a more complete selection of taxa is considered. However, these results suggest that skull volume can be a reliable predictor of carnivore and feeding ecology in a larger, more broad selection of taxa. The third and final question was, are different locomotor and dietary categories statistically different in body shape parameters? So in front of you is a phylogenetic tree, which is of all the species that have been tested, and each of them have been put into a locomotor group. The reason that in front of you you're seeing locomotor rather than diet is because that we found that most significant differences in limb length parameters were due to locomotor type and not diet. So we found that phylogenetic ANOVA reveals that the femur, shank, pes, humerus, forearm, forelimb, hind limb and average leg length are significantly affected by locomotor style. Now, results using non-phylogenetic statistics yield many statistically significant differences in body shape between various locomotion dietary types. However, when phylogeny is considered, most differences lack statistical significance, which together with high values of Pagel's lambda suggests a strong phylogenetic signal in the dataset. Most remaining statistically significant difference were found in the forelimb, which has been highly modified during evolutionary transitions in locomotion. So in front of you, you have a graph of the humerus length of different animals regarding different locomotor types. Anything with a red circle above it is significantly different to active flight animals, and anything with a pink square above it is significantly different to soaring animals. So for example, you'll find that flying animals tend to have a relatively long humerus, while aquatic and semi-aquatic animals have a short humerus. So identifying significant differences between locomotor type and ecological niche may allow quantified body shape analysis to be applied to extinct species for the prediction of ecology when behaviours cannot be directly preserved. So on the right, you have a phylogenetic diagram of locomotor type. So in the red circles, you'll see birds and bats, which are your active flight animals. And on the left, you have a phylogenetic tree showing humerus length. So the green, yellow, and into red colours are those animals that have a longer humerus length per body mass. And preliminary work shows that active flight animals converge on similar relative humerus lengths. Regarding the forearm length is very similar to the humerus, whereby active flight animals have significantly longer forearms than bipedal striders, aquatic, semi-aquatic and quadrupedal striding species, whilst soaring species have significantly longer forearms than aquatic, bipedal striding, quadrupedal striding, semi-aquatic and arboreal animals. When looking at the forearm length, again, we can see that the higher values tend to be in your active flight animals, so your bats and your birds. In particular, bats tend to have longer forearms than your birds do. And preliminary work shows that active flight animals converge also on the forearm, but not as much as the humerus length. So we show that relatively simple comparisons of the forelimb and humerus bone structure can distinguish between most locomotor groups. This technique is easily applied to many fossil specimens and has the potential to shed new light on the locomotor behaviour of extinct taxa. For future work, I'll be investigating specific evolutionary transitions where there is a major evolutionary shift from one locomotor or dietary category to another. An example of this is the evolution of flight in birds, and looking for patterns here of convergent evolution. I'd like to acknowledge everybody from Liverpool University and everybody in the EMB and outside that have helped me with this research. And thank you very much for listening.